So I'd like to begin by acknowledging the traditional owners of the land upon which we meet today, the Wurundjeri people of the Kulin Nation, and pay my respects to their elders past and present, and any elders from the communities who may be here today. It's my great pleasure to introduce Phil Hodgkin, Professor Phil Hodgkin today, from the Immunology Division. Phil began his days in the bush of WA, ended up doing a PhD with Kevin Lafferty, who, for those of you who don't know, Kevin Lafferty was didn't fear to speak his mind, and there's a Lafferty debate named in his honour at the Australian Society of Immunology meetings. And it was there Phil got interested in T-cell decision-making, and that's been a constant theme throughout his career. He then spent some time at DNAX with Maureen Howard, returned to John Curtin University working on quantitative signal integration. He was one of the first adopters of, many of you may know CFSE, this technology, Australia's second biggest invention since the Hills hoist. It allows you to track division from cells, and Phil was one of the early adopters in that area. He came to WEHI in 2002, became division head in 2006, and he's continued to work on those same questions. And Phil, I think in 20 years, he's going to get embarrassed when I say it. I think in 20 years' time, when we look back on the immunology history of WEHI, Phil will be one of the kind of main figures we talk about in continuity from Mac Burnett and Gus Nossel, because he has a different way of thinking about biology that, if we're honest, not everyone has a completely different angle on thinking about things. And you'll see from his talk today, I'm sure, that his work is in T-cell biology, but whether it's cancer or development or neurons or lungs, if we think about, and he was into single cell biology before it was cool. And if you take away, I hope you take away from this talk that if you think about things on the cellular level, then you can perhaps understand biology in a completely different way to what you otherwise would have. So anyway, no pressure, Phil. But please come ahead and give us your presentation. Thanks.
Wow. First of all, can you hear me? Yes? For some reason, my voice has gone a bit soft, so I'll try to speak loudly. Thank you very much, Charlene. I am embarrassed by you saying that. I sincerely hope that's true, but I would have to say at the outset that, yes, I have dabbled in modelling and trying to do things quantitatively in immunology and think somehow outside the box. And I can tell you that it hasn't been easy. There's no real textbook that you can go to how to do this. There's no mentors really that you can go and learn at the feet of. Very much for me, it's been a matter of making it up as I go along, inventing the problems and then trying to invent the way of solving the problem and then hopefully finding a solution to the problem, having come up with the question, which hopefully makes it easier. And so my intention today is to just give you an overview of the value of models in immunology. I'm going to go back, some distance in time. I'm going to start by really asking the simple question, what is a model? And of course, there's a classic answer to this. It's some sort of mechanical description of some system, preferably a complex system. Building models is integral to the scientific method, of course. Any scientific investigation aims to develop a model of processes, and that model will embody the principles of some theory or hypothesis that you want perhaps to test in an experiment. And as you continue in this scientific investigation, your models hopefully will become more and more sophisticated, perhaps transitioning from a qualitative prediction of what might happen to a very precise quantitative prediction. At least that's what happens in physics. In biology, the quantitative part gets much harder, as we'll see. So let's just put that into a little bit of context, because this to me is a great inspiration for how to approach problems in science. It's the classic example of how to work your way through a hard problem. And it took many centuries, of course, to work out the motion of the planets around the sun. what's going on there. Of course, we start off with Aristotle and the Greeks, with the Earth at the centre. And then Ptolemy started measuring roughly, and things didn't quite fit, so you had to have circles within circles. So this is Ptolemy here. Copernicus eventually puts Earth in the centre, Sun in the centre, and orders the planets out, but it's still everything in circles, it doesn't quite fit. Tycho Brahe builds this big observatory and measures things very carefully and shows that, in fact, these models that have been emerging over hundreds of years just don't quite work. And it's left to Kepler, really, to look at all the data that Tycho Brahe had and gouge out what's called empirical laws. So these aren't based on some sort of deductive reasoning. They're just gouged from the data, and they will explain what's happening. So this is the ellipse, and how much of the ellipse is covered in a particular time, all worked out by Kepler. And of course, then enters Galileo, starts studying just falling bodies. He couldn't measure with he used his heartbeat as his watch, and he would drop bodies, but he couldn't measure them very accurately, so he started rolling them down inclined planes, working out the single properties of objects in motion. And he came up with some brilliant ideas that eventually, of course, inspired Newton to deductively solve this problem. And to the point now, of course, I'm an astronomy nut. I have a telescope that I just dial in my date and my GPS coordinates, and it knows exactly where I am, and it points to exactly anything in the sky at any time. We can use our phone to position ourselves in Mars and look at what the solar system would look like at any time in the future. So the equations, just a few simple equations, solve this problem completely and distill all this data into everything we need to know. It's an amazing feat. Okay. So I'm going to tell you, did I skip a slide? I did skip a slide. So the greatest achievement, and I'm going to tell you this because it's going to be relevant to my talk, I think the greatest achievement of that whole sequence of events is Galileo's demonstration that you could take a complex system, so bodies subject to multiple forces, and separate them and study them in isolation, and then use a little bit of math, in fact just a tiny bit of algebra, to put them back together, and assuming they're independent, was able to predict the consequence of the two things acting together. So this separating into components and using math to put them back together is the principle, really, of modelling and reductionist science. It's had a huge influence on the development of science over time. Here's Galileo here, rolling objects for the king. Okay, so 5 stories of modelling immunology. I may not get through all 5, but the first one, Pauling and the pesky graph, Lafferty and the second signal, CFSE and the cellular calculus, calculating destiny and division randomness and fate. So let's get into it. Pauling and the pesky graph. Here's Pauling, Linus Pauling, and I put this slide to illustrate just how famous Linus Pauling was in the United States. He ended up on a stamp for his work in sickle cell anemia. Of course, he defined many aspects of the chemical bond. And at some time in the 40s, he got very interested in antibody. And to give you a little bit more of that backstory, here's the late 1800s, Emil von Behring discovered that immunity was evoked in serum after being exposed to some infection, diphtheria or tetanus here in these guinea pigs. He could transfer that immunity in serum, thereby discovering antibodies. Landstein, has shown here, was interested in, well, how many different antibodies could you make? And he's a chemist, and he made organic compounds that had never existed on Earth before. And he said, can I make antibodies to those? And in the 20s, late 20s, he published that, yes, you could. No matter what you make in the laboratory, you can make antibodies to it. So that made it seem very unlikely that you could have pre-existing antibodies or code for pre-existing recognition of anything like this. So Pauling stepped in and presented his theory of how antibody must work. His idea was that the secret of the specificity was in the molecule itself, that all antibodies had the same sequence from a modern perspective, but that what happened is when the foreign material came into the body, it went to some site where antibody would wrap itself around the three-dimensional shape, the surface of that particular antigen, that foreign material, make a version and be cast off. And then another antibody would make a complementary version and be cast off. Make another, it's like a factory, moulded on the template of the antigen. So this is the mechanism of the model, hold and fold. Okay, this theory is in the 1940s. This was tremendously popular. Pauling took out patents on this idea. He was really pushing this hard. Now, if you apply your scientific method, and I'm really addressing the students here, can you predict the consequence? How should that antibody accumulate in the serum if it's made in this way? And this is the process of applying a model to data, or making a prediction from a model. So Pauling's presented his model, he's told you how he thinks it works, and you can predict how that antibody should accumulate in the serum. Anyone want to have a guess? That's right. I'll pretend I have. The prediction is that it will accumulate in a linear fashion, because the factory is just building one molecule at a time, so it'll just accumulate in a linear fashion. And here's the first data for antibody production from late 1940s. It looks linear, right? But carefully note, this is a log scale here, and this is a linear scale here. So this is a log linear plot. A straight line on a log linear plot indicates an exponential relationship. So having been exposed to antigen, the appearance of antibody in the serum goes up exponentially. Who did this experiment? This was done by McFarlane Burnett here at the Walter and Eliza Hall Institute. By 1948, he's the director of the Walter and Eliza Hall Institute, very interested in the immune system, but of course a world-famous virologist at this point. But he did this experiment, and as a consequence, because it didn't fit the model that Pauling was promoting, he rejected that model. He wrote a book with Frank Furner, Production of Antibodies, 1949, pointing out this flaw in this model. He presented some idea of what an alternative might be. It's completely wrong. It's actually quite bizarre. He had another couple of goes over the ensuing years, and it wasn't until 1957, provoked by a few other studies, that he published this paper, which was the landmark paper. It was really the correct solution to the problem of how antibodies could be made to this huge number of different antigens. This is his famous clonal selection theory. I'm not going to go into it in detail because I hope everybody here is well familiar with this theory. But just to point out again that he published this paper in this newsletter of the Australian Society for Advancement of Science. He was president at the time, no peer review. He wrote it on a weekend. The first page is all introduction. The second page, every paragraph starts a new field of science. So antibody will be a receptor. The receptor drives activation of a cell. Each cell will make only one type of antibody. You grow up, it will stimulate proliferation. Memory is due to the increased number of clones. If you break down, then you'll have autoimmunity. And importantly, there must be a randomizing mechanism in the DNA that he didn't know anything about for combinatorially creating the millions of possible receptors. All of these turned out to be true. Notice the paper that appeared after this, probably greatest paper in immunology, certainly one of the greatest in biology in the last century, is followed by some practices of the Pacific Island peoples handling and storing foodstuffs. So, not a well-known fact. Okay, so let's just draw a few lessons. So here we have imaginative model building at its best, tested by experiment, used to reject a very popular theory at the time, to rethink in this classic fashion that you saw with the development of the Greeks and the understanding of our position in the universe, and then start again and go over again. The other point to make is that despite Pauling's great fame and authority, Burnett, here in Melbourne, the underdog in this race, could ignore his ideas just from the slope of a line because he understood the quantitative aspects. So quantitative prediction is a stern test of any model. That's an important way of conducting your scientific method. And that's kind of the theme of if I get anything across to your students today, is that making quantitative or semi-quantitative predictions is a very strong test of your understanding of what's happening. So Burnett left a trail of wrong guesses, but eventually the exponential increase to him implied cell growth. And that's what gave him the clue. So that simple line on a graph gave him the clue he needed to start thinking down the line of clonal selection theory. And of course, once published, this new theory unleashed enormous interest and activity in further investigation here and around the world. So lesson number 2, Lafferty and the second signal. Now, here's a little timeline here. We've got Burnett and his clonal selection theory here. He didn't give many instructions about what would be required for the lymphocyte to get activated. And Burnett, of course, only knew about one type of lymphocyte that was going to make antibody. And it was Jacques Miller who, not too long after, showed that there was another type of lymphocyte, a T cell responsible for cell-mediated immunity. And a little bit later on again, Miller and his student Mitchell showed that these T cells and B cells had to cooperate to make antibody. So this lovely simple theory of Burnett was getting more and more complicated. And in fact, Jacques suffered the slings and arrows of people saying, you're complicating our immune system. Please don't. Jacques here. This is a great contribution, but it already said that filling in a theory of the immune system is going to be hard. It's going to get harder and harder. The first go at explaining what might be happening here was a theoretical idea from Peter Brecher and Mel Cohn. They published what they called the two-signal theory, and their idea from first principles that what this communication's about is protecting self-tolerance. So we can't just allow this deletional idea of Burnett to take away the clones that are self-reactive. That's likely to go wrong. We need a second opinion. So their idea was that every activation event required a second opinion, that there's two independent antigen recognition events and as a fail-safe against responding to self. So that was their two-signal theory, like a binary switch. One signal gives you tolerance, two signals, you can become activated. And into this environment now comes Kevin Lafferty. Kevin didn't start out being theoretical. He had nothing particular, no axes to grind here. He was just interested in this question of tissue recognition, graft rejection. Why is tissue? Why do so many T cells respond to foreign tissue? And could graft rejection be prevented if we knew the mechanism by which T cells were recognizing? So here's the model he's working with, the Burnett-Medawar model, right? That the T cells will respond to antigen, become activated and start dividing. So just antigen recognition alone should activate the cell. So the foreign material could be a virus or a bacteria, but it would activate the cell like this. Histocompatibility antigens could be the foreign material and activate the cell like this. So simple model, simple prediction. The frequency of responding cells would increase as the material becomes more and more foreign. As the tissue becomes more and more foreign, you'd expect to have a greater and greater immune response. And it was that Kevin tested experimentally. And he used a system of graft versus host reactions in the membranes of chicken eggs. So he put, chicken on chicken, and they're different at MHC, so they get a strong reaction. But duck is weaker, and I think this is pigeon, no? Goose, hardly at all. Put mouse on there, nothing. So the further you went away evolutionarily, the more foreign they should look, but the weaker and weaker the response. So here again, the opposite of what you expect, an astonishing contradiction of this simple model. So Kevin turned to in vitro studies of the mixed lymphocyte reaction. Now he's turning to mouse. You can put lymphocytes from one strain here, say CBA, with a second strain bulb C here, and mix them in a culture, and they'll proliferate like crazy. And you can measure proliferation by tritiated thymidine uptake, very high sensitivity. And what he discovered was that if he changed species, the stimulating cell was a rat or a guinea pig, the response weakened, just as he saw with those chicken responses. If he used non-hematopoietic mouse cells like fibroblasts, the response was very weak. If he took the supernatant out of these cultures, mouse on mouse, and put them in a mouse versus guinea pig, then it went like gangbusters. So he came up with the idea that he called a two-signal theory. And the idea was that T cells needed a second signal. He called the term co-stimulator comes from Kevin Lafferty. That second signal is the co-stimulator. His idea is here, that your responsive cell here, think of it as a T cell, recognizes antigen, it gets the first signal, but that somehow triggers the presenting cell to make a second signal here. And it's only with the combination of this signal plus this signal that the cell is able to respond. And this second signal is not antigen specific, but it's species specific. It involves a receptor here, and this has to bind this to provide that second signal. So if you change species, the response gets weaker and weaker. That was his explanation for that weakening. Okay. So as a consequence of challenging the model, coming up with a new model, he made a bold prediction. So here's a tissue transplant. He imagined that within this tissue transplant there are cells capable of providing the second signal. He showed that only hematopoietics, we would think of them as dendritic cells now, but are able to provide these second signals here. And they carried along in the blood or within the graft. They're not the bulk of the tissue. He imagined that they come out of the graft into the lymph nodes, activate the T cells. The T cells, once activated now, can recognize the foreign material anywhere in the graft and attack it and reject it. And his thesis was if you could remove these cells that initiate the response, it would be able to be accepted by a foreign host. And he was able to show this by taking, this is thyroid grafts, a week's culture in high oxygen, enables that graft now, this is after a week, highly rejected with lots of lymphocytes. After a week of culture and transfer, this is a very healthy thyroid graft. And they could survive after that culture period for months. And if you just injected a few spleen cells into these mice, these grafts would rapidly reject. So if you provided that second signal now, they would rapidly reject. So a vindication, again, of challenging the orthodoxy, coming up with a new model, and making a bold prediction. Okay. What lessons? Again, imaginative model building, tested by experiment, rejecting the current dogma, rethink it, and following, if you like, this classic scientific method in order to come up with a new model. And again, despite now Burnett and Medawar's great fame, Medawar was like the doyen of immunology in the UK, and Burnett, of course, here. Their fame and authority, Lafferty, ever the iconoclast, as Charlene pointed out, is the underdog in this race, but he trusted his simple experiments and created a major advance as a consequence. And again, to make this point, a quantitative prediction provides a strong test, a very strong test of the prevailing model. Okay. So now, let's turn to some work from my lab. This is CFSE and the cellular calculus. So Lafferty's legacy was this question. Okay, it's fine to tell us there's a second signal, but at a molecular level, what is that second signal? And Charlene mentioned that I went to work with Kevin around about this time, 1982. And he was already on the trail of trying to identify a molecularly clone what that second signal might be. It was initially thought to be IL-1, then IL-2, then eventually IL-6, CD2, CD28, and this becomes the sort of popular favourite. But in sequence, many, many molecules were shown to influence T-cell activation. to act as, if you like, a co-stimulator. So Kevin was driven mad by this, in a sense. And what his simple concept was very complicated by what is the co-stimulator. And of course, this got even worse when these molecules could be knocked out in vivo, and very often, even though they had big effects in vitro, tenfold differences with and without, in vivo, much smaller effects, if any, quite often. Very specific to a particular immune response. So this was like a paradox. The paradox is kind of given lip service in textbooks. So here's Janeway's immunobiology, two-signal activation. Here's your T cell seeing antigen, getting a second signal here by the most popular second signal CD28 here. And as a consequence of this activation, the T cell will make this autocrine growth factor interleukin 2, and that will drive proliferation over here. Doesn't answer the question of what all those other co-stimulators are doing and how they might all work together in vivo. And if you take away CD28, quite often your immune responses proceed just fine. So this is a complex pharmacological cell biology problem now. How do the lymphocytes sum all the signals and make decisions? And in particular, just focusing on this decision between whether you should respond, is this foreign or not, and if you respond, how strongly should you respond? So this is really the question. Now, the in vitro co-stimulation assays based on counts per minute were very sensitive. As I said, many different co-stimuli could change this to this by adding something, CD28, IL-6, IL-1, IL-2, all of them will do that. Now, if you want to ask what's actually happening there, how is it achieving that enhancement? It could be that more cells are dividing. It could be that the same cells are dividing but just faster. It could be that less cells die before they start dividing, or less cells die as they divide. There's all sorts of possible combinations of way that could be explained. And if we really want to understand how these signals are acting, we need to understand what's happening behind that simple bar graph. And I venture to say that just looking at counts per minute, it's impossible to give that answer. And this is where this division tracking dye has become so valuable. Carboxyfluorocene succinimidol ester. And Bruce Lyons, who was a postdoc with Chris Parrish in the John Curtin School in Canberra, and they bought all the fluorescent dyes they could from molecular probes. They were labelling lymphocytes and transferring them in vivo to see which ones would work to allow them to track cells into different sites. And Bruce used this particular dye, transferred it in vivo, and recovered the cells from the spleen after a few weeks and noticed these peaks. And he realised immediately that these cells were dividing, that the dye he had labelled the cells was stable and it was diluting faithfully between the daughter cells. So you could see cells in different states. Okay. Bruce Lyons was in the lab next to me. I was interested in division tracking because I was interested in B cells changing into antibody secreting cells and isotype switching, and I'll tell that story a little bit later. But I wanted to tell you in the context of this co-stimulation story is that now for the first time we have a way of measuring division rate, and by carefully counting the cells, the death that's going on in those cultures. So if we take a cell and it divide, we know it divides 3 times and we recover 8 cells, then we know we've got everything. If we only recover 4, then some must have died somewhere. And so this is the type of accounting problem that I applied to this co-stimulation question. So by now, to do this, I'd moved to the Centenary Institute in Sydney, and Amanda Gett was a fantastic student who did this work with me. Now, over here is a typical T-cell proliferation. T-cells have been labelled with this dye, stimulated with anti-CD3 and saturating IL-2 now. Okay? And at this stage, no CD28. We'll get to that. And you always see this type of pattern. So some cells here already at 60 hours, still undivided, division 1, 2, 3. 12 hours later, cells, you can see they've progressed. 12 hours, further hours, they've progressed even further, but always spread across divisions. So some cells have obviously divided faster in some way than these cells. We can set gates and just plot the number of cells in each generation over here. So this is division number versus proportion of cells in this direction. So we get these curves and you can watch them moving through and you can see how symmetrical they are. Okay. Now we can fit a normal distribution to these and just plot the mean division number here versus time, division number versus time here, and we get this straight line. And from this straight line, we can calculate a few things. We can watch the intercept with division 1 here to work out the average time the cells are taking to enter their first division. And from the slope of this line, the steeper it is, the faster they're dividing, the faster they're moving through that. So from the slope of this line, we can work out, once they start dividing, how quickly are they dividing. Now, once we know how quickly they're dividing once they start, we can take this distribution here, which is in division number, and convert it to time. Because it turns out that the bulk of this variation here arises because the cells vary in the time they take to enter their first division. Once they start, they tick through pretty systematically. Okay? So we've taken this data, done a little bit of analysis here, and generated a time to first division distribution, a mean and a variance for that, and an average division rate once you start dividing here. Just 3 numbers. Those 3 numbers I've used here in Excel to recreate. This is just done in Excel using those 3 numbers to recreate the exact data here. So this is data, and this is the recreation based on the numbers produced in this very simple model. And this is overlaying the proportions based on the model. And you can see by playing with the mean and the variance, you can play around and get this to fit. So now we can ask the question, we've got a nice little analytical tool to dissect the process of division, how division is working. Just 3 parameters. So over here, we've done this experiment again, with and without CD28, this co-stimulator, this second signal. And you can see that if you look carefully, the cells here are divided more than here. But what is the pattern? It turns out that CD28 doesn't change the slope of these lines. So once they get started, they're dividing at the same rate, but it's changed the time they took to enter their first division. The variance the variation in the time to that first division doesn't change either. So co-stimulation here with CD28 was affecting proliferation in this way. We could do the same thing with another co-stimulator that gave good enhancement of proliferation, IL-12, and that didn't have any effect at all on the proliferation parameters. The effects of IL-12 are entirely on survival as a consequence. Interleukin-4, though, has 2 effects, the dotted lines here. So with and without CD28, it reduces the time to first division and increases the slope. So once they get started, they go a bit faster. Now we can play this game, this Galilean game. We've separated the variables, we've put them together and asked how do they combine? And it turned out just by adding the difference, so the effect of CD28 increases time to first division by this amount, the effect of IL-4 increases it by this amount, put them in together, add them together and make a prediction about what it should be, and that's precisely what we saw. Time to first division here, 2 experiments, very close. Subsequent division times as well. So the effect of combining the 2 signals was predictable in a very simple arithmetic way. So lessons. CFSE allows quantitative accounting of division and death. Applying it to T cells, we can take a quantitative view of co-stimulation and see that there's a gradation of influences. We can change the rate of division and the rates of death. There's not a binary switch on or off. I didn't show you, but this simple model is hypersensitive to small changes. I'll come back to this in another context. Also, didn't show you, but we looked at how cell death intersected with the division, and we could show that it looked independent. So we could treat death and division separately and put them back with algebra. And for this reason, I acknowledge Galileo for this idea, which I stole in this paper, in the acknowledgements of this paper. So, and this approach has inspired many subsequent studies along the same lines of complex signal addition. Alone, together, can you predict what's going to happen? And we're doing that over and over in our lab in different ways, in different contexts, looking at different fates. And two more examples in the next slide. Okay, calculating division destiny. Now, hopefully you heard Julia Marchingo talk about this story, so I'm just going to skim through the details, but I think it's a nice story, building again on this co-stimulation story. So, with T cells, we're stimulating them, we did all those analyses, we're putting in IL-2, so they just continue to divide. But when Marion Turner, a student in the lab a few years back, did this with B cells stimulated with CpG, a toll-like receptor, she found that they divided, and then just stopped. And if they're wild type, they would just stop and then start dying. And if we did it in BCL2, transgenic B cells, they stopped but didn't die. They just stayed there. So the number, they would divide, and stop. And we termed this behavior division destiny, that the cells seemed to know how many times they were going to count through and then stop. And they returned to being small quiescent cells. It's like an automatic response. Edwin Hawkins, student also at the time, started filming these cells. CpG-stimulated B cells don't stick together. You can follow them individually. And we saw that exact same, that behavior. We could watch that behavior. Cells, here's 3 cells. This cell grows, divides, and then then no further motivation to divide and eventually dies. This one divides 3 times, and this one divides 4 times, eventually stops, and there's no motivation to divide and eventually dies. And over again, even though when we track individual cells, we see some variation in how many times they're going to divide, even though we're giving them the same stimulation, within a family, we almost always saw this behavior. So this cell divides once, twice, 3 times, 4 times, symmetrically, and all the progeny stop dividing in this generation and go on to die. Okay. So what's happening there is that the number of divisions before returning to rest varies, but families have the same pattern. This first cell passes the information to all its descendants, how many times to divide. So this is what we call this cell's division destiny. So we're seeing this founder effect on the destiny. So here's our sort of our cells in our well. Somehow secretly they're passing on a division destiny number to each of their progeny to when they're going to stop. Okay. So we've never seen this behavior in T cells, but it occurred to us that this destiny would be a nice explanation for some of that paradox of the co-stimulators that Lafferty was confronted with. Maybe we could get some more clues about how co-stimulation was working. And this became part of the PhD project of Julia Marchingo. As I mentioned, she has talked about this previously, but maybe you weren't all at her talk. So with T cells, as I mentioned, usually you don't see this behavior. They continue to divide until they reach the limit of your ability to track divisions with this die. It's about 7. They all start stacking up. But of course, T cells make this growth factor interleukin-2. They also often are stimulated with lots of co-stimuli. So we stripped out all the signals we could. We stripped out the IL-2. In fact, we titrated, we blocked it and added in. We blocked the mouse IL-2, the endogenous IL-2, and added in human IL-2. And, And you can see with high concentrations of IL-2, they continue to divide. With low concentrations of IL-2, or in fact, no IL-2, you can see they divide, they stop, and they start dying, just as we saw in the B cells now. Divide, stop, and start dying, just as we saw in the B cells. So this same behavior, this automatic behavior we're seeing. So here's a nice film version of the same thing probably much more appealing than watching those little graphs. But this is a T cell that's just divided. It's got the Fookie reporter, so red G1, green SG2M. Just look at the synchrony of this little family of cells. And we see this over and over. So it's divided 4 times. We have this family of 16 or I think it's 32 cells. None of them divide again, and they've all gone red at this point. So immensely symmetrical behaviour passed on from generation to generation. Okay, so Julia went on to use division tracking. We're not using CFSE anymore, it's this violet dye, but the same principle. The cell over time start dividing, eventually they stop and they start dying. We can represent that here, accounting for the division expansion. If the cells proliferate they'll just, and not die, they'll just follow this line here. 100% of our starting cells will follow this line. If they stop like this, they'll stop here and then they'll start dying. So we get this crash plot. And this line here tells us what the division number, the destiny of this, under these conditions, what the destiny was. So what affects destiny? Lots of things. A T cell receptor affinity will add to how many times the T cells will divide. CD28 adds to how many times the T cells divide in a dose-dependent manner. CD27, another co-stimulator, adds to the number of times cells divide. The cytokines, interleukin-2, adds to the number of times. IL-12 adds, IL-4 adds. Not everything adds. IL-6 weekly, 21 very weekly, 10 interferon, nothing. So then we go back to this trick of asking, okay, that's all the single effects. Let's start adding them together. How do the cells integrate the information? Can we determine a quantitative rule for how multiple signals are integrated? So here we've got the situation titrated. So this is just the T cell receptor stimulation, and this is with a low dose of CD28 here, anti-CD28 here, a little bit of a co-stimulation. We're adding about 1 division. Here's with anti-CD27, another receptor, adding about 1 division, and then we can add them together and make a prediction. If they simply add together the 2 arrows, here to here, we would expect this to be here. If one was dominant, we would expect it to be here, or they may be synergistic. And remarkably, over and over again in different combinations, we saw the 2 just added together in this additive way. They're just added. So you can change the cytokines, you can change the mixture. It always works. Okay. Now, the real important point about this is when you add divisions, of course, every time you add a division, you get a 2-fold increase in your cell number. Add 2 divisions, you get a 4-fold increase in your cell number. So there's a very sensitive effect of adding divisions in this way. So all these co-stimuli are integrated into the cell and adding divisions. So here's 3, for example, here, CD28, 27, and IL-12, giving us this addition here. And this is just showing the actual cell numbers as we're adding in the co-stimuli. And you can see we've gone from a weak response here to a 10-fold greater response just by adding in 3 weak additional, 3 signals, adding 1 extra division at a time. And this is putting it on a log scale, a linear scale, so you can see it's quite a big difference. And you just remember those co-stimulation plots from, no co-stimulator to a co-stimulator. Okay. So this, I think, is another insight into this paradox of co-stimulation that Lafferty was confronted with. And so here's 2 elements to the solution. First of all, to the question of what is the second signal? Why can't I find the second signal? The answer is there is no second signal, essential. That there's many inputs can influence T cell proliferation by adding the number of times those cells divide. And so, if you like, an infinite number of combinations, if you just set yourself the task of getting 10 divisions out of your T cells, which would be a big immune response, you could get there. There's probably 10 different co-stimuli that can add to division. So many different combinations of concentration and combinations can give you that larger response. So there is no essential second signal. There's an integration of signals that just sums into the overall response. The second point is, how is it that in those in vitro assays, 10 different things can look like they're essential. You take them away or add them, you get a 10-fold, a 5 to 10-fold increase in the number of T cells. How would that be possible? It doesn't make sense quantitatively. And I think this is what's happening. So if we imagine a situation where we've got our T cell receiving some combination of co-stimulatory signals shown here in these coloured arrows, and each one adds a number of divisions here. So we're getting to about 9 here. Now, if we took away any one of these, we took away the blue one here, or the pink one here, or the red one here, we would reduce the number of divisions overall that it underwent simply by about 20%. We would just reduce the division number by 20%. And if we model the consequence of that in a simple model, we start with the blue line here proliferating and coming down. That's this situation. If we convert to any one of these, in other words, if we take any one of those other co-stimuli away, we convert down to here. And now if we looked at early, we wouldn't have much of an effect. So what do immunologists do? They always titrate the time and find the point at which they get the maximum difference. And that's what goes in the paper. So this happens over and over. It's a problem, I think, in scientific investigation today, because we're compelled to maximise the effect. And it makes it very hard for theoreticians to try to fit everything together, because you end up with this situation like the co-stimulation paradox, where you have 10 things all contributing a 10-fold effect. And I think this is the answer of how that works. Okay. And of course, the other paradox is, why doesn't the in vitro system match the in vivo system? And that's almost certainly because of this autocrine growth factor interleukin-2. It builds up in the medium very quickly, and as you can see here, this is different IL-2 concentrations progressing the cell number. It has a massive effect on the division number. It's a very potent effect. And so IL-2 is, if you like, subverting your ability to see what's really happening in these in vitro cultures. If you take it away, then your in vitro cultures match pretty well what happens in vivo. So lessons. Again, hopefully imaginative model building and quantitative methods allowed us to solve a long-standing problem, and that the disconnect as well, which is important for moving forward, between in vitro biology and in vivo can be resolved. Also highlights the uncomfortable secret that many immune studies are optimized to maximize an effect. Now, you're going to have to, as students, you're going to have to confront this. You're going to be asked to do this to promote your paper. And again, quantitative prediction is a powerful attribute of the new model, even in vivo. And I haven't shown you, but this is a lovely experiment in the paper. Please read Julia's paper, showing that we can increase affinity or add in IL-2. The two effective affinity and IL-2, we can make a prediction of how they would add just in this simple way and predict the consequence of the two additional signals here, and it fits beautifully. So the same principle, the same approach that we've used in vitro, we can apply in vivo. And this is really the first time we've managed to be tremendously hopeful for the future, that we can continue to improve our models to make accurate predictions in vivo as well. So I was pretty sure I wouldn't get to be able to take you through division, randomness, and fate. So I just want to sort of give you a flavour of it. So, of course, here the problem is the adaptive immune response. It's been talking about proliferating cells, but of course they differentiate. They have enormous potential to become different types of effector cells. B cells can make all these different types of antibodies. There's all these different types of T helper cells, for example, Th1, TFH, Th2, Th17, Th9, Tregs. And somehow the lymphocytes start by integrating information from dendritic cells, macrophages, and neutrophils, lots of inputs into their decision-making that somehow influences what they're going to be and how many they're going to generate of each type. This is another problem of signal integration. How do we go from there? How do those signals integrate to drive the response here? This is, if you like, is the same problem we've just been talking about with additional dimensions, additional fates. So what is the source of that heterogeneity? The conventional view is that each of these fates would be driven by a cytokine or some particular instruction. It's a personal instruction to that cell to make IgE, if you like. I think I wrote that in there, personal instruction. Every difference is driven by signals, personal instructions given to each cell. So this map of T cell differentiation, Th17, make IL-17, but they'll need IL-4 and IL-21. So each arm here, there's a cytokine driving it in that direction. And there's no doubt that's true. Those cytokines will influence the behaviour. But it turns out, I think there's another much simpler explanation of what's happening, and it greatly simplifies our task of modelling this process, I think. And that these differences arise automatically. They're intrinsically programmed, they arise automatically. Yes, those cytokines can influence how rapidly they become that fate, and we can design our experiments to maximise the look of that cytokine inducing that fate. But very often they're not essential for that fate. They're just changing the rates. So how could that work? So it turns out these reduction experiments of trying to reduce all the variables and stimulating cells and getting them to divide, nevertheless, a great deal of heterogeneity remains. Cells, which is consistent with some sort of cell-intrinsic programming of multiple fates. So I've talked about the evidence of destiny. So the idea that the cells integrate signals and then just divide through a number of times and then stop. So the whole, if you like, mini immune response is programmed into that first cell. It doesn't have to be told at any point to stop or to die. It's all playing out as a consequence of the initial signal integration. Fate changes with division. That's something we noticed very early with the CFSC. This is a very early experiment giving B cells, dividing, spread across these divisions. And if we looked at those B cells switching to IgG1 with the cytokine that induces them to switch to IgG1, IL-4, or IgE, you can see that there's no switching before division 3. And then only ever about 50% of the cells switch. And IgE takes about 5 or 6 divisions, and then only ever about 50% of the cells switch. So it's not true that the cytokine will drive the fate. There's other factors at play, one of which is the cells seem to have to divide in order to do this. The same is true for becoming antibody-secreting cells. Here's division along here and generation of antibody-secreting cells. With WEHI and Steve Nutt and Lynn Corcoran and Dave Tarleton, we worked out that there's an emergent program, and Gordon Smythe, there's an emergent program of RNA expression changing with division number. Okay, so we went ahead and imaged fate to ask the question, how do they interleave? So how does becoming an antibody-secreting cell, how does isotype switching, how does division, and how does death interleave in these cultures where everything's happening simultaneously? And I know I'm late, but we imaged thousands of cells under the microscope here, all counted by the heroic J-Zo here. So this is the results of the division times, death times, differentiation times, switching times. Amazing correlation between siblings, division times, death times, differentiation times, switching. So two siblings, almost molecular kinds, have the same fate. But nevertheless, enormous heterogeneity. We also saw division and death being highly correlated, or division and differentiation being highly correlated across clones, suggesting that there's some, at face value, there'd be some mechanical connection between those two fates. Now, we didn't necessarily believe that, so we went and tested this with a very hard model. And I can't tell you this I'm just going to have to give you the quick answer. That correlations can occur as a result of censorship. So if every cell is assigned a division time and a differentiation time, randomly we get a sort of pattern like this. But if when a cell divides, it resets its differentiation timer, then we will never see these events because the division censored it. And as a consequence of that, this random pattern becomes correlated. And so we applied this idea to the data. We imagine there's a distribution, every cell's randomly selecting a division time and a death time and a differentiation time and a switching time. Each time they divide from these distributions independently, and ask if, as a consequence, we could fit that data and see those correlations between fates that weren't division and death, for example, or division and differentiation. And that turned out to be true. Observed times of fit were fit to the censored distributions in the model beautifully, and it explained over here these unusual correlations at the right level. The data and the model fit were within 95% confidence. So I'm just going to wind up. Steps to accurate model of immune responses. The immune system is amenable to model building. The B and T cells respond with an automated divide, stop, die program. Statistical tests indicate division, destiny, and death machines are independent in the same cells. I believe it's reminiscent, it was to me, of the Galileo-Newton actual calculus. And as a consequence, we can model proliferation survival very simply, as I showed you, with 3 numbers or a few more if we incorporate death. B cells show strong evidence that further fates, differentiations, becoming antibodies secreting or switching, are independent and in competition with division and death as well, and therefore you can account for striking, enormous amount of single-cell heterogeneity as a consequence, and that this, I believe, is a strong, powerful framework for a multidimensional cellular calculus for multiple signals integrating and affecting multiple different fates. Molecular variation and expression levels likely underlies this emergent heterogeneity, matching the population to the molecular pathways, and that's something for the future. So here's the future dreams that I just wanted to finish on, that the clonal selection theory will become quantitative and predictive. We're not there yet, but I think we're getting there. That the 2-signal theory of qualitative decision-making that's in the textbooks now will be replaced with quantitative model of precise signal integration and include the laws for how each signal affects the outcome alone and in combinations. And that these principles of cellular calculation can be codified into computer-based tools for data fitting, simulation and prediction, and that our ability to predict cytokine effects, genetic changes, drug effects, as a consequence of these models, will bring about a revolution in the practice of immunology, as new accurate predictive models always do. And I sincerely hope this all happens here, and that some of you may help. Okay, many people have helped on this journey. I've just listed them here. I've tried to point them out as we went along. Thank you.
Thanks very much, Phil. We've got time for a couple of questions. So please fire away, students.
I'm pretty sure Fabian's not a student, but...
You had the video of the dividing cells and they turned red, the divider went back to green. Why did they all come together with this complex signaling that makes them all divide at the same time?
We're pretty sure there's no signalling because in situations where the cells are mixed, they still divide according to their family if you track them. I think the theory is that the stage is moving a lot. It's actually returning back to that. It's taking like 50 movies at once over a few minute period. And so it's moving, it's jerking, and it may not be completely level. So when cells round up ready to divide, they're not anchored in any way. They're not moving or they're not attaching. And so they're just kind of billiard balls. And we think that they just sort of, by inertia, are being pushed to one side. That's our theory. Trivial explanation, but maybe there's something more to it, but we haven't found it.
Any other questions? Here we go, one here.
You mentioned that all of the signal integration that you observed was additive. Is there a reason that you think that everything has been additive and you haven't seen any dominant effects or multiplicative effects?
Yeah, I'm sure in some situations we will, because you can imagine 2 receptors sharing an exact same pathway or very early coming to the same point. It's hard to imagine that will always add, or at least it will saturate at some point where they both can't continue to strengthen the same signal. So there will be situations where they add. We're certainly, you know, a little bit like the co-stimulation cheat. We're cheating by reducing our signal, so that, which we have to do in order to be able to track the division numbers before they hit 7. Now, it may be if we were close to the saturation level of CD28, then CD27 wouldn't add because it's already closer to the saturation path. Those things, I think, will be informed as we know more and more about the signalling pathways that are being shared as well. So it's a very good question. Obviously, I think the principle is that they can operate independently, but it would be very surprising if that's always the case. It's true. Yeah.
In the interest of time, I'm sorry, we're going to have to finish. There is provided lunch for the students in the boardroom afterwards. Phil, thanks very much for inspiring us with the butterfly effect of immunology.